Hello, welcome to Microbiology Journal Club. This is our Microbiology News Week, where we talk about some of the latest microbiology news, and then we talk about some of the interesting papers that you might want to delve in a little bit deeper in next week. I'm Fazal Lam, and
And I'm Danny.
yeah, and we're here to talk some science, talk about all things great that are small.
Yes. Yeah, I think it's been a good time talking about these papers over the past few months. I mean, it's definitely like a lot of we've talked a lot about COVID-19. Um,
Yeah.
our backgrounds are we were doing our PhDs in infectious disease microbiology, and I think it's come out in the papers that we've chosen to talk about further, right? Like it's nice to talk about those mouse models and think about how they were used and all the animals that they are going through when they're doing those
Well,
studies.
yeah, we're quite interested in the underlying aspects of disease, of how COVID
Mhm.
works, and we don't necessarily get that from human studies. It's very rare.
Yeah,
I mean, things like cell studies. We've looked at bat cells, we looked at various animal
yeah,
cells last week, and looking at just the underlying molecular, like how does this tiny machine make so many lives miserable? That's kind
yeah,
of
absolutely.
how we've been working at it. I mean, we've also been looking at the animal.
And
I mean,
oh,
part of the thing that's great is that we've also been looking at the flaws and things. And animal models, there are lots of shortcomings that people have to take into account before we decide to assume anything from them. Um,
absolutely. Yeah.
yeah. Uh,
Yeah, so if any of that interests you, like you should definitely tune in and send us a tweet or something about something that you're interested in, and we can incorporate into our discussions.
yes. And I guess now we can start with the news.
Mhm.
Yeah. So this week we've had some. So I think this past 2 weeks we've had this news on convalescent serum. So,
Yeah.
yeah, we've talked a little bit about it. It's often used in the vaccine studies we've talked about as like a comparator for how well as good is the antibody response we're getting, and they compare it to
Yeah.
convalescent serum.
Yeah, so convalescent meaning someone who was infected before,
Yeah,
previously an infected person.
yeah,
Yeah, and I mean, like that's actually, at the beginning of vaccinology, right before they had vaccines, this was the way in which people immunized each other, right? There was a big system of um, convalescent serum, sometimes even and from animals as well, right? They would just immunize animals with things, and then they would take out the horse serum, and then you would get horse serum, right, pumped
right,
into your blood.
yeah,
Um,
with
and that's because, we make antibodies. Those antibodies are in the serum, and so you can transfer those antibodies. Passive transfer
yeah,
is one of the ways people talk about it in the research literature.
and it makes sense from a logical perspective because if someone's fought off the disease, they must have had the right antibodies. Therefore, if you put those antibodies into someone else, surely it should work.
Hmm.
But
Yeah, so this news was the FDA approving emergency use authorization for convalescent serum,
yeah,
and some controversy, right, whether or not the evidence in which they were using to approve that was sound.
yes. We're going to be talking a bit about the paper that was used to justify this, which was Joyner et al. So let me
Hmm.
see if I can pull that up quickly.
Effective convalescent plasma on mortality amongst hospitalized patients with COVID-19.
Yeah. So this paper, I mean, just looking at the medRxiv page, it looks okay, and then suddenly, unlike most medRxives, there is a ton of comments in the comments section that are often very long and very involved because
Uh-huh.
this has caused quite a bit of a what we call a stir in science.
The technical
The
term.
technical term is rumpus, I believe, because when this was released, people read through the paper and found that there were some issues that. This study was performed in quite a lot of people, and it was based on so they had like two groups. A group that was given convalescent serum before a 3 day mark, and patients that were given convalescent serum after 4 days. Now, I don't know if you already spotted the problem here, but the question we want to know with this convalescent serum is how much better does it improve treatment outcomes? So you need
Right.
to have a untreated control group, which they did not have. This was
And
a
I think it's a product of the fact that this is not like an interventional clinical study.
mm,
It's not like they enrolled people who had COVID-19, identified ones that they could give serum and not, there was always the use. People were using serum, like even at the very beginning, before this emergency use authorization, people were talking about serum.
Yeah, I mean that's the thing that surprised me. I didn't think that because I didn't think there was anything stopping people from using serum.
Yeah,
Um,
yeah.
and I know that there are other studies. So I know the recovery trial is still looking at whether convalescent plasma is going to work or not.
Hmm.
And there are there's like a couple of like other studies going on this time, but it's odd that this was like the first one to come out, and it also immediately got like a ton of people looking into it and asking like because the thing is we don't really cover observational studies too much in this
Hmm.
because observational studies come with a lot of like extra things that you need to take into account.
Yeah.
So
Well,
all
because there's all sorts of because people aren't enrolled ahead of time, right? There's all sorts of things like it could be that the same types of patients are being given serum, and so you're seeing just like also the bias coming from the doctor's orders
yeah.
coming out in here. Yeah.
So, like, if a patient's like very sick, the time they get the serum might be very different to the time if they're well. So, the way that
Hmm.
treatment groups are assigned in this aren't random. They are dependent on so a certain so that means like a certain
Yeah.
patient might get it.
Factors that aren't even accounted for necessarily. Like they only do the analysis afterwards, so they try to control for it all afterwards.
Yeah,
Yeah,
which is
adding layers of covariates and stuff.
yeah. So yeah, that's another like issue with this where you have to do the analysis because usually for any random RCT, any experiment, you want to plan out what analysis you're doing before you do it. So, but
Hmm.
because if you do it afterwards, then that means you can actually choose which analysis will make your results look better,
Hmm.
which is something we
Yeah,
always want
and you
to.
might not. Yeah, yeah, you don't necessarily disclose those ahead of time. That's the whole point why we see those clinical trial registries and stuff, right? It's trying to keep people doing those trials honest by saying, please disclose all your methods. And we've talked about that before when we were talking about um, that big UK study, right? It's
Yeah,
like they have to publish all these methods ahead of time, and they were very careful to build in some ways in which that they can change them, but they accounted for it all ahead of time, and then
yeah,
afterwards you still see some variance, but without doing that, there's even more sort of like flexibility in the way that people analyze the data.
yeah. There is the issue where you can pick your favorite analysis. I think it's called hypothesizing after results are known, or harking, where you
Oh, parking.
it's like the equivalent of the I don't know if you've heard of the Texas gunman, where the Texas sharpshooter, where you fire an AK-47 into a garage, and then you draw your target over the three nearest shots and say that
Yeah,
you've hit your target perfectly. That's kind of
right,
like so you spraying things randomly and then deciding your target afterwards, which
right.
is is kind of bad practice and makes it very hard for
Yeah.
people to.
Like there are right statistical methods get there are statistical methods that are supposed to take that into account, but it's still not like the most rigorous way of testing those things, right? This multiple
Yeah.
hypothesis testing stuff. There's little corrections people do, basically raising the bar of what's significant.
Yeah, and a lot of those really involved statistics usually go into observational studies because a big thing that happens that everyone has to be aware of is that what you're testing for, there could be a third option, a third thing that you haven't thought of affecting everything. So you need
Hmm.
to always be careful about all the other unknown confounders.
Hmm. Yeah, I find this is odd to me that it is such a large news splash. I guess. I mean, I think maybe it's not odd because I understand there's all this like political stuff happening. I think with like how is the FDA being used as an organization in the states. Um, but I mean, like the data is here, right? We can look at these things, and obviously people have been using serum
Yeah,
in certain cases. So I guess I'm not really sure. I guess the EUA is supposed to help more people use serum, or maybe it's supposed to encourage companies to start making serum for others. Like I don't really understand like what the use of the EUA is here, and yeah, why it's so hotly contested. I understand the paper being contested. I think to me that's like regular science, right?
mm,
Like we're having the discussion about it. We're just seeing it, and we have so much attention on it because it has this air of political issues sort of around it as well.
yeah. I think the main issue because I think everyone who like is criticizing it said, well, I mean not everyone because the thing about convalescent serum is that it is a mix of different like antibodies from different patients
Mhm.
and I think I might talk about this before, but it's also from patients who not necessarily from patients who have fought off the disease very well. Because if you're in
Mm.
hospital, compared to like you're not necessarily have the antibodies that are fighting off disease because you've ended up in hospital and in a severe condition. Whereas the thing about coronavirus is sometimes they do have this antibody response that actually impedes the clearance of disease. So that's something that
Mm,
people have to be careful of when looking at convalescent sera and looking at the
sure.
results from that. And that's the thing that's going to be hard because with convalescent serum, you take all these different cases that are from different people and pull them together. So you'll get quite a large variation in sort of reactions. And so it's not always very clear that it's going to be. So it's not the same as if you got one specific antibody and give it to people. This is a
Yeah.
whole mix of different
Yeah.
things. So it's not.
I mean, that means there's going to be variance in the product too. Like
Mm,
if right different different places that make it are going to have people will have different results with it.
yeah.
Um, yeah, it's not good to think of this as like a a a a a, a drug or like a standard therapy, right? I think it even with emergency use authorization, it's still something sort of on the outskirts that like it may or may not work depending on who's giving that serum.
Yeah. And yeah, the amount of like there are so if the viewers want to take a look at some of the critiques, they're on the medRxiv website if you want to see. They're like peer review is happening in action in the comment section. Uh,
Mm-hmm. Mm-hmm.
so I think we can move on to our next news story perhaps,
Sure.
which is going to be the Calgary et al. So this is something that you want to talk about. So I'll let you take it away.
Yeah. Kelger et al. Which one?
Calga.
Oh yeah,
Sorry,
Cowger. Yeah, yeah,
Calga.
yeah. Yeah. So this published paper, which is kind of cool, a case
Yes.
study about. Sorry. This is not that.
So
I thought this is something. This is published. This is talking about racial disparities in COVID-19. And I picked this out. I got it from the John Hopkins. They have like a service. We've linked to it before where they read papers and they do a little overview. And it's well known that there's some racial disparities, right, in the way that people get treated. And here they're saying that that could be underestimated because of the way in which minorities are estimated, like in the regular demographics of the country, is what they were saying here. And so it's kind of interesting. I mean, it's a very simple point, right?
yeah,
That speaks to statistics that we always hear, and kind of shows you that you have to dig into that data to really understand what's going on, right? And in this case, they went in and they look at like weighted with the known. Right? This is the major observation. Like when they weight based on certain proportions of minorities in the country versus unweighted without those proportions, they see different, they see different things come out. And from that, they concluded that, yeah, it's an underestimation
yeah, no,
with the way that we've been talking about things.
yeah, that's really
Yeah, so
interesting.
it's a pretty short paper, right? It's like it's only a single figure
Yeah, so
that kind of shows that. Like, I think that's why like I wanted to be. We're probably not going to talk about this. We're not really epidemiologists either to dig into this, but I appreciated that it was very understandable. Like that sort of simple sentence, like I guess that gets expanded on by looking at this. Um, yeah, they were able to communicate this sort of issue.
yeah,
And I
so
think it's
yeah,
important. It can be important for money too, right? Like I like the funny thing is like what do you do with this information? I think is like always the big question. And it can inform things like policy and it can inform things like showing the limitations of the way people are analyzing things, right? Like you can show like need, right? There's more need in
yeah,
certain communities. I think that's probably a really important thing that this type of research can
yeah.
help with.
And I think it's very important to take those into account because I know, like definitely in the UK, certain communities have been much more badly affected by this outbreak. I mean,
Hmm.
lots of Asian medical professionals have like been really severely affected by this, and
Hmm.
also communities, and it's important to understand because like while we're scientists and we're focused on petri dishes, there are all these other sociological issues that impact this
Yeah.
coronavirus outbreak that we're not necessarily that we haven't been as good at talking about.
Yeah.
And
And I think an important thing to remember too is like this is also like these people are also science, right? There's the social scientists, the
yes,
epidemiologists. It requires this sort of nuanced understanding of statistics, right?
yeah,
Like that like if we were better trained, we could jump in and we could look at this figure at a glance. Like right now, like the figure to me is sort of like,
okay,
okay, I trust what it says, right? But it's always like this, like diving into the figure and saying like they really tried to illustrate what they're seeing, right? As experts in the field.
yeah, it's because again it goes into things like reporting standards and across different, yeah, it's and yeah, it is
Yeah.
interesting about because again, if depending on like what area you report, the portion of the
Hmm.
population is going to change. So it's
Right. Right. So using like a blanket statement of like this is how, right? Like basically if you use a blanket estimate of everyone in the country, right, had this proportion of minorities exist, that you're going to have issues because the reporting of the people who got sick is not just a blanket amongst everyone. It's like it's based on regions, right? And so like I think that that's what they were doing when they weighted things is that they looked they went in depth, right, to each specific area and looked at the statistics there and then correlated that against the people who got sick or
mm,
the people who are dying.
yeah,
Yeah. Anyways, I thought that was an interesting thing to pull up.
yeah, I think so. Thank you for pulling that up. Um, I think that brings us to the last bit of news, but I think we're going to linger on this because it is quite interesting, which
Hmm.
is the COVID-19 reinfection by a phylogenetically distinct SARS coronavirus 2 strain confirmed by whole genome sequencing.
Yeah, Toe et al. Right.
Yes.
And this one was published right in CID. This I thought we were talking about. This is published in yeah CID, but there's also an unpublished account as well that came out just a few
Yes.
days
I
ago.
think I've got
Uh,
the. Let me.
Tillett Tillett et al. Genomic evidence for a case of reinfection with SARS-CoV-2.
Okay, hon. Wait. Yeah. Tillet. Uh, oh, wait a minute. I should go to our Zotero, which is over here. Right.
Yeah, but I mean, both of them use the same evidence, this genomic evidence, which I think is the only way that you would be able to tell, right, if someone got
Yeah.
reinfected. Because there could be reinfections with non-genomically distinct, but that's going to be a lot harder to show because how do you know the one that got reinfected first is different from the one afterwards? And you'll only know through um, yeah, you'll only know through the genome if something is different. So in these two papers, they sequence the strain before and then they sequence the strain after. I think the difference is that the patients in these two, one got sick both times and
Yep.
one only got sick once.
So that would be Toe et al. Their patient only got sick once, and then they had
Hmm.
a less severe infection next time. Whereas in
Yeah,
yeah,
and I,
with
I think
Tillett et
until it
al.
they got sick twice.
Yeah. So yeah, they yeah, which is unfortunate for both of them, and
Yeah,
it's
but I think what's the implication is that it's about spread, right?
yeah,
And oh my goodness, I feel like so people show like on um like what is all the excitement about antibody testing? That's
mm,
I always ruminate over this, right? Like what's the excitement about antibody testing? And there's no studies that tell us, right, like which antibodies are the protective ones
yeah,
and what even happens when you have those protective antibodies. But there is excitement, at least in the States. I've seen the excitement. Like all these clinics are like, we have antibody testing here. Like, come get your antibody testing. And it's just unclear what you're going to do with that information. Like, I can understand from the surveillance point of view, right? Like, okay, we have this approximate amount of people who may have been exposed before. But I mean, even then, the studies that we've been reading about some pre-existing antibodies to these antigens, right? Like just in the baseline
yeah,
population, is complicating that image. But I secretly suspect that some people believe that I got it, so I'm okay, and I can do whatever I want now.
yeah,
Um, and I don't think that I never really I didn't think that was the case. And seeing these two papers is more. evidence on that side that like we still don't really know what's going on. Like you should still be taking precautions, even if you have antibodies, because you could either get the disease again, or you could just be sending it out to give it to somebody again, right? You could be a node.
Yeah, and I think there was a story about like the antibody titers being decreasing a lot faster than people were expecting. Now that could be uh, due to many factors, but I mean the idea is that maybe the antibody responses aren't necessarily perfect, and
Hmm.
that or that I mean there's a suggestion that memories for this isn't so. I mean for other coronaviruses there have been studies where they have shown that people can get reinfected with the same strain. So
Yeah, I
um,
mean, I think that's what's thought with the seasonal ones, right? Like they don't
yeah,
change that much from year to year. It's just that like there's a new susceptible group of people and they get some of the cold, some of this cold.
yeah, and so it could have impacts for coronavirus and vaccinology because I know that there was like some
Yeah.
earlier studies in rhesus macaques that came out where they found that rhesus macaques could not get reinfected, which was like a big beacon of hope for people.
Yeah, yeah, absolutely. I mean, I think that like the basis of understand like yeah, the basis of understanding convalescent serum, right,
Mm.
even, right, is
Yeah.
saying that like, oh, like this it works in some cases. And that's not to say that it doesn't, right? But
Mm.
there's also now we now have two examples of where you can get reinfected, right?
Yeah.
And cause disease again. Yeah, that rhesus macaque one we also actually have in our Zotero. It's animal disease models. It's I at
Yeah.
least thought it was.
And yeah, I think the main thing that I want to recall with the rhesus macaques is so they don't get the same sort of disease that humans do. They get something that's a lot more mild. So it could easily be that they are, since they're already less susceptible, that sort of reinfection would be a very rare event that you would never see in a study that has involves rhesus macaques because
Sure.
they can only use so many. Whereas these are probably quite rare because we can only see two cases, and maybe the the outbreak goes further, people look back, maybe they can find more cases like this. But I
Well, I think that I think that that's one of the things I was trying to say is that it's a strange. We can't know whether people would be reinfected with the same virus over and over again, right? That's like
yeah
it's really hard to get that information. So like these are the examples we have of one type of reinfection. It could be that that different virus is somehow right interacting with that person's immunity in a certain way. Like the
yeah
immunity that they produce like isn't good enough to deal with that variant because in each case, like it is a variant. I don't know if they went in and they looked if those variants were meaningful.
so
Um,
I think they did
I know
so
in Toe they show like the spike. They are distinct spikes,
yeah
but whether or not like that means anything, right? Like they can just show us that from the genome. We don't know whether right the antibodies are binding differently to those two different spikes.
yeah because I think for Tillet they look at the sequences and uh, they're not supposed to be that different.
Hmm.
But let me check. So because I need to jump down to the figures.
Yeah.
Ooh, yeah. So yeah, I mean, yeah, there are definitely some differences, and so they do the whole genome sequencing. So I think with Toe they
Mhm.
only look at the spike, whereas with Tillet they look at across all the genes, and they find like some differences.
Yeah. No,
And
I think they did whole genome sequencing too in Toe.
oh right, yeah, okay,
Yeah, the uh, reference genome. Yeah, I think they did both.
yeah, okay.
But like the figure in tow, like they laid it out nicely to show you like what those differences are. It's a bit harder to see until it like what those different. I mean, they did case A variants and case B variants. I guess you see them across these two orphs. But if you don't remember what these orphs are,
Yeah.
oh yeah, Spike. So yeah, they have the same variant of Spike here actually between the two. I assume that's what it means, the red line. But
Uh,
anyways, it's without going deeper, I don't think we can say too much. Just that there were differences in the genomes, which could mean functional differences, but they don't do any experiments to tell us that those genome differences really do correlate to a functional difference. All we know is that, yeah, it's possible to get reinfected again. Stories. And
Yeah.
again, this is the thing that you could show through genome sequencing, but it doesn't rule out the case that you could also be reinfected by the same strain. We just
Yeah.
don't know because like that's really hard to show.
Yeah. Because you have to be able to like say without a doubt that someone has cleared the disease completely, and it's
Yeah, and
very
then got it.
very
Yeah, and got
yeah.
it back. In this way, it's easier because they know like the initial disease was the majority this one, and then the second one is the majority another genotype. So like that gives you that resolution.
Yeah, I mean, I would have liked some. I mean, the problem is since it's only two people, you couldn't do it that advanced antibody characterization that we saw in other papers, where you'd be able to
Mhm.
characterize what these and the antibodies were actually looking at.
Well,
We
I mean, I guess you could. It would be again, it would just be a lot of right because you can take the people's serum and you could take their blood cells and you could expand them out. And you could do that
true.
epitope mapping. I think it's just I think the reason why it's not done is that's just a really involved study.
Yeah,
And if you think about the type of lab that's going to do genome sequencing versus the type of lab that's going to do a bunch of like immunology tests, I think that they're different. And so my guess
yeah,
is that like these people just don't have the capabilities. And like you have to consent that person, right? You're like, did they take enough blood from the first infection,
yeah,
right? To be able to do that type of characterization in the second infection? I think that would be the biggest limitation now that I think about it. Right? Because you want to know what happened in the first infection. You need blood to look at those cells.
yeah.
Did they take enough blood in the first infection to be able to do that type of analysis?
So yeah, you're right. I mean, because also I think that once you have this result, you want to publish it because all that other data takes a bit more time, whereas the
Yeah.
readout from a genome can actually be very very fast. Uh,
Absolutely, yeah. I'm sure that this one was just like the doctors were like, "This is bizarre." Like from the clinical side, they're like, "This is strange." Then they like were like, "Oh, great. Well, we've sequenced this." So like then the people who did the sequencing dug for the files and then did the comparison. They're like, "Fantastic, publishable."
yeah, yeah. Yeah, and also, so I think that's everything for our normal news segment.
Hmm.
So I guess I'm gonna go on to our vaccine news. How are we doing in the COVID-19 vaccine Olympics? Well, this week has actually been relatively quiet. Pfizer have come out with their new vaccine. Well, the vaccine that they really want to go with. So the one we analysed in our previous study was the what version one, which was just with the RBD receptor binding
Yeah.
domain, whereas this one is with the whole spike protein. And looking at the studies, briefly going over it, it seems that the main benefit of it seems to be less side effects, less kind of adverse reactions. And
Hmm.
so that's why they've gone with this whole spike protein rather than just the RBD. Uh,
Yeah. Yeah,
let's
so
see.
this is nice because
Yeah.
this is their preprint now that's talking about the whole spike protein. Previously, the only thing out there was some of the other candidates that they had. So at least they're giving us more about how this one's going on.
Yeah, and last time I missed some news on a phase two trial on a different whole cell virus that people are testing. So we've got Sinopharm and Sinovac,
Hmm.
who've both published phase two trials, and we've got Adenovirus 5. They've published their phase two trials. But aside from that, not really much more on the whole vaccine side. But well, I mean, a lot of the big ones are going in phase three right now, and so there is that hope. Everyone's hoping that we can get something very soon, which is
Hmm.
unrealistic, I think. The
Yeah, I
idea,
mean, realistically, what sort of news might we expect? Like, I think we'd only be hearing new drugs that appear that are going into a certain phase of trial because it's going to take a while for things to go through like their current phases, right? And we might see interim reports, which again wouldn't be
yes,
published papers. They'd be probably like press releases, my guess is, right? Like showing some things. And I don't think we'll see those either until like people are pretty confident, or maybe if they need to sell something to the investors.
yeah. I mean, Pfizer have two other vaccine candidates that we haven't heard anything about yet. So
Hmm.
that we could always get a surprise with those. We've got
What were the other two that Pfizer have?
well, I remember. Well, they didn't actually say it in their clinical trial paper. All I know is that those two vaccines exist. So they're testing four different vaccine candidates.
Oh, just
They
the other B whatever
BN
B3.
something.
Yeah,
Yeah,
I
I
see.
forgot. All I remember is like the one, two, three, and the four. The end last parts.
Yeah.
Um,
Yeah, that's true. They could be going forward with those as well. It's potential.
yeah, we don't really know too much about that. What other things are there? Uh, yeah, that's basically it for vaccines. I mean, there's been a couple. There's always new people coming out with vaccines and small. So I've created a small section of Zotero to like filter out filter out things based on whether in that phase. So I think on the Zotero file, let's see if I
Oh
can.
yeah, vaccine papers tracker.
Yeah, so I'm still new at this Zotero, but I've basically been like adding like what's in phase one, phase two, what phase and phase three, and what's been done in primates.
Sure.
So I'm going to try and keep adding that, keeping that updated to so that eventually I don't want to do like a big old comparison between some of these things.
Yeah.
Uh, but yeah, that's it for vaccine news. So quite short this week. Yeah, and
All right, so like let's look at some of the meatier papers that we might be able to dive into next week.
yeah,
Um, what do we got? Oh, this distinct conformational states of SARS-CoV-2 spike protein,
yes,
Kai et al.
yeah, Kyle. This is one I quite. So I came across this while researching last week's paper, and it's one that kind of because because last week we were talking very much about that fusion reaction that happens between SARS and the membrane, and this
Hmm.
actually goes into the protein structure of how that happens. So that's
Hmm.
why it really caught my eye,
Yeah,
and
structural biology. I see lots of structures.
yeah, so because yeah, I
Oh, and
guess
it's sort of telling us how the fusion is going to happen as well, right? It's diving
yeah,
into that.
that's why I quite like it because it treats like it's not just looking at the structure; it's looking at the structure in motion and looking at how it acts in vivo, which is just like uh, which is brilliant. I quite like it.
Yeah, I mean, it's really fascinating stuff, and maybe it could help. Yeah, so this one, oh yeah, this is a good one. Um, I think I'd picked this up before. So this is related to things like the crystal, the original time the crystal structure was, right? Like this is a extending on stuff, right? Like at the very
Yeah.
beginning, there was a structure, not a crystal structure, a
Mm.
cryo-EM structure of the whole spike. But now they're finding a moment, the shape when it's in another conformation, right? Because they're dynamic. You only ever see like snapshots of things.
Yeah.
So that's cool. Yeah.
So yeah, it's quite cool. Quite feeds into that story, and also cool. We're getting quite interested in the whole life cycle of the SARS-CoV-2, and seeing like this very interesting part of it was something that I thought was
Yeah.
quite intriguing. And
Absolutely.
yeah, let's see. Um,
Okay.
okay, let's see
Yeah,
what
so that's
other.
like a structural biology one.
Yeah, uh, what other ones are there? There's feline coronavirus drug inhibits the main protease of SARS-CoV-2 and blocks virus replication. So this one has hit the headlines a little bit about like, oh, this coronavirus drug that was for cats can probably help humans. And
Hmm.
looking at the study, I kind of tend to class it more in the structural biology rather than in treatment because what they
Mm.
actually do in it is doesn't really speak much to whether it will treat well in humans because they do a lot of in vitro work and looking
It's
at
like
like
what we did.
cells.
It's sort of like what we talked about last week, right?
Yeah,
Like, oh, here's a pharmacologic intervention, and they look at it binding, but if they haven't tested it in something physiologic, then it's still a little bit far away from thinking that
yeah.
way.
I mean,
But we do learn the similarities, right? Like, oh, this pocket is the same between the two versions, um, cat and human.
yeah,
Yeah.
and I think it does speak to that really interesting thing of uh sorry I'm getting a slight echo the interesting thing of finding out how things work by breaking them, and this is what this coronavirus drug does. It breaks so it targets a protease that we haven't talked about before. We mostly talk about spike protein, but there's this M protease made by SARS-CoV-2, so it's an entirely new kind of aspect of SARS we haven't looked at, and we can and yeah, we can also have that treatment perspective that is quite people quite interested in. Oh, Sorry, Danny. You don't. I can't seem to hear you. Is your microphone working? Um. Oh, okay. We've lost Danny. He's uh yeah. Can't really hear you on stream, unfortunately. Um. Let's see. So quickly send me a message to see whether I can fix this. Um. Because we are getting some feedback from you, but I.
How about now?
Yes, perfect.
I just toggled my microphone.
Oh, okay. Okay.
I was just gonna say that this links a little bit to the paper that we did, right, like last week, right? In that like we were talking about looking for small molecules that block the spike, and then
Mm,
here's another small molecule blocking a protease. And I think that we have like some in our backlog in the small molecule thing. There's a structure-based design of antiviral candidates. Oh, targeting SARS-CoV-2 main protease.
mm.
So yeah, so we also there have been attempts to put something in here as well. It would be nice. We could this would be a nice way to feed into that one.
Yeah,
We could talk about both.
yeah. We could do another doubleheader episode where we look
Yeah,
at small
about
drugs
the
for.
smart drugs for the protease. Yeah.
Yeah. Let's see what other things do I have that are structure related. I've got
Furin
oh
cleavage site. Oh.
yes, furin cleavage site. That's right. So uh, again, going back to last week, we're looking at how cleavage is quite an important process in the SARS infection. So, and I've been hearing like talk about how furin cleavage is quite important to SARS-CoV-2 infection. So this
Mm-hmm.
is why I got quite interested in this. Um, Again, this is in a cell-based study, but it looks at a mutant lacking the furin cleavage site, and then basically seeing what happens. So again, we've got
Mm-hmm.
a key SARS protein that's slightly broken, and we can use that to figure out how SARS works and what happens.
Furin is a host protein, right? Furin is
Yes, I
host.
believe it is. Yeah. Um, so I mean, last week we talked about host proteins like TMPR, SS2, and
Mm-hmm. That did the
uh,
pre-processing.
yeah. So yeah, this is so that's why I was kind of interested in
And this
looking
is
at
also the
this.
spike. This is also spike.
Yep. Also Spike,
So
and
it's that pre. It is about the pre-processing actually.
yeah.
um, yeah. Oh man, I want. Is furin? We didn't really talk about this, but they used two inhibitors in the last time, right? They used one that was specific for um, TMPR, but then they also used one that was like for Cat B or something, Cat A, something
Yes.
like this.
Yeah.
I wonder if this falls into one of those families.
I mean, it might do. So yeah, it
Hmm.
might. And they also do like they go from like cells into a hamster pathogenesis model. So
Okay, they
they
use hamsters.
yeah hamsters, which we've talked about before that they're slightly better than the mouse model because they actually have human ACE2, so they do get that disease.
Hmm.
Um, and yeah, the only difference with hamsters is having the infrastructure in place. So again, it does it still takes out like cell structure and then goes that one
Oh,
step further.
the hamster icons they use in the figures are so cute.
Let me check that out. I have to put that on stream now. This is going to be the thumbnail for the stream. If they have to be
Yeah.
cute now. You've
Oh, they are cute. They don't, yeah.
yep, they are cute.
Right? They're like really like I would have like stickers of those for
Oh
sure.
man, yeah, they look so anime. Yeah, they're
Got
yeah,
some nice
totally. I'd
shade in
definitely buy a
there and
plush of
everything.
those. Which makes me feel worse now. That makes it. I mean, if you're gonna have it in a paper you're affecting hamsters, don't make them so cute.
Oh yeah.
Oh, so yeah, that that may have
Yeah,
sold
this
me on this paper, but we there are other
it's
papers we
interesting.
can be looking at. Let's see. ORF9. There's so uh, SARS-CoV-2 ORF9. So Lulu Han et al.
Hmm.
SARS-CoV-2 Orf9 and Orf9b antagonizes type 1 and type 3 interferons by targeting multiple components of the RIG1 slash MDA5 dash MAVS, TLR3 dash
Oh,
TRIF, and I'm getting to the end of this if it kills me, and the C gas sting signaling pathways.
nice. Congratulations.
Yeah, I really broke a sweat saying that. It's just that
Sound like a real
MDA5
immunologist.
Mavs really got me. But yeah, this is interesting because signaling pathways are like one of the key things of immunology, all
Mhm.
that communication that happens, and seeing how SARS.
Yeah, we've seen like the panels of cytokines and things, right? Like, and that's the stuff that the cells are spitting out, right?
Mm,
They're spitting out these cytokines. Those cytokines are binding to things on the other cells. But then don't forget, like inside the cells, then binding of those cytokines are gonna trigger phosphorylation cascades and all sorts of things inside the cells. So that's what we're seeing here with all these things. But RIG and MDA, those are intracellular, aren't those intracellular
yeah,
like sensors?
I think they
Those
are.
are, yeah, those are binding like things in the cells, which I guess makes sense because that's like that's the idea, like it's surveilling for RNA inside of the cell, right? Like in humans we only have RNA,
Yes.
right? That makes our proteins. So if you see RNA that binds to itself in a strange way, like a viral genome, that can be an indicator of something bad is going on.
Yeah, completely. And yeah, this is an interesting part of like seeing the innate immune response to COVID,
Mhm.
because I think that's something we didn't really talk about in the we probably could talk about a little bit in the news. There are innate viral responses, and so
Yeah, yeah, exactly. Like this is the stuff. Um, yeah, this is the stuff that basically in evolution, that's what I was saying, like viruses have that double-stranded RNA genome, right? Like we don't
yeah,
see that in humans. So like evolution just says if you see that, it's time to heighten the alarm a little bit.
yeah,
Yeah.
and that's I guess that's one of the reasons why like the mRNA like kind of vaccines could, if you have like a double strand in there, that could cause the immune that you don't have
Yeah,
to have that much of an adjuvant because you can trigger that kind of innate response
exactly.
that leads to the
Yeah, exactly. I mean that could be the thing, right? It's like it makes the protein and it also triggers this like yeah, the signal. Yeah, TLR3 is the one for double strand is the toll-like receptor for double-stranded RNA.
yeah.
So
So yeah, and if SARS has a way of like
dealing with
interfering with,
that, yeah. So
that's
this is
interesting.
accessory proteins. I mean, I definitely, I'm feeling this idea. I want to talk about another SARS protein, if that's protease or ORF, because it's like we
Yeah,
talk so much about Spike, and like we know Spike is going into all these vaccines, but like to understand disease, like so many other things are happening.
yeah,
It'd be nice to flesh out that picture in some way. Yeah, so this is some accessory protein, ORF9b.
yeah. So yeah, we're kind of spiked out at the moment, but having
Yeah,
this,
yeah,
but this
yeah.
is yeah, this is interesting because it teaches us not much. It's not just about the SARS-CoV-2, but also a little bit about the immune system and how that works.
Hmm.
So we're covering two topics that I'm not too good on. So it's
Oh,
great. We can learn lots more.
and they definitely have a bunch of bar charts with with different cytokine stains.
Yeah, this is going to yeah. So let's see what the figures look like. Yep, the figures.
Ah, but they do. We never really talked about this, but subcellular, like co-localizing as a
Mm,
way of indicating detection, right? Or do that.
right.
Oh, some Western blots.
Yep. Some.
Lots of Western blots.
Yeah. Lots of Western blots. I'm just automatically like scared every time I see a Western blot. Just like what's.
Oh, you'll like these. These are just like they just cut out the bands. You don't even get the
Yeah.
whole gel.
Yeah. Don't know. Can't see the whole gel. So.
Gotta take it at face value.
Yeah. So yeah, this could be a reasonable one to cover. I mean, no pictures of hamsters, but I think we'll survive. Uh, yeah, and
Yeah.
let's see what other things are
So
there.
I think the thing to just to throw in that an extra fact right now, like innate immune system does impact adaptive immune system, right?
Mm,
Like there are
Yeah.
these two arms, but they do impact each other. We sort of hinted to it by saying adjuvant as like the bridge, right? Like that's the
Mhm.
dark magic of vaccines is that they have to tickle both in some ways to make the response.
Yes.
Yeah.
Yeah.
And so we know the adaptive, that's the antigen that we're putting in. We want adaptive to respond to it, but then we also do other stuff to try to get the innate to say, like, this is also important.
Yeah. And so this covers topically it covers both boxes.
Hmm.
And yeah, so I think this is a strong candidate to discuss further.
Yep.
Um,
You also have another intense immunology paper, I think, that Kaneko
yes.
et al. Loss of BCL6 expressing T follicular helper cells and geminal centers in COVID-19.
Yeah, now this is kind of an interesting one because what they do is they take people who have passed from COVID and looked at their like look at I think lymph nodes. They
Hmm.
take like samples of lymph nodes and spleens from people who had the COVID-19 infection and look to see whether there are like B cells or other things in there. And they basically what they find was that they seem to have lost their germinal centers, which kind of indicates that they had an impaired immune response,
Hmm.
which
I think this is so like this isn't okay. So when I first entered grad school, I considered myself like somewhat knowledgeable about the immune system. Like I knew things, like I had taken courses in university about it, but I had never heard of geminal centers before I came
germinal
to,
centers. Oh,
yeah,
oh
germinal
wow.
centers. Yeah, before I came to grad school, I guess like that just wasn't in the breadth of immunology that we got in regular undergrad. Um, but yeah, anyways, it was like so. This is I really enjoyed it because it gave me that like spatial resolution, right?
Yeah,
Like when you think about the immune system, that like oh man, it like matters. Like even in the space of just like a single lymph node, there's like all this stuff going on,
yeah,
right? Like where is that cell going? Who's it associating with? And that's like part of the developmental cycle, part of priming the adaptive immune
yeah.
system, right?
Uh, it's like when your lymph nodes start swelling during like an infection. That's partly due to change in structures in lymph nodes of all this. So germinal centers are where antigen presenting basically sort of happens, where and sort of then expansion of
Hmm.
adaptive immune cells, if I recall correctly. But the thing about it is that
Yeah, yeah.
the more I learn about immunology, the more I realize I know nothing about immunology. So
Yeah. But again, like so reading this paper would be like brushing up our understanding, right, of these sort of like early phases of the immune response or like
yeah,
maybe it's also like antigen presentation in the immune response. T follicular, that's the other thing, helper cells, right? That's very much antigen presentation, priming, um,
yeah.
and stuff like that.
So I mean,
BCL6 though, isn't that like a oh no, never mind. I've just heard that in cancer. I actually don't know what
I
it
mean, it
is.
probably would be
But so
because all the everything that's important in because BCL6 is something that's important in I think uh, in various immune cells. But again, I'm not too familiar with that. But it wouldn't surprise
it's
me if it
probably
was also
proliferation. If things
yeah,
that are linked to proliferation always end up linking back to cancer, right? Like if it's normally signaling your cells to grow, then cancer will definitely have a broken version where it's like always on.
yeah. That's the thing that about cancer. And that's I think we talked about why cancer research is so important because those proliferation signals are always important in other places, such as right here.
Hmm.
Um, yeah. So
We got a lot of co-localization and pretty pictures in this one as well.
yeah. I mean,
And seeing that structure, like I mean, just looking at these, I'm like, you get that sense, like, oh yeah, this tissue is has these foci of cells.
yeah, I think that. So I'd quite like to tackle this because I think it is an even peg in terms of the ORF9b and the loss of BCL6.
Hmm.
And so BCL6 has a lot of the figures look so much nicer,
Hmm.
but ORF9 it does tell us a bit more about coronavirus. So this is very much like so I like the study because of the pictures and the story it tells, but it also has that kind of observational aspect to it where it is very
Yeah, because they
patient.
look at some human samples afterwards. Are they biopsies? Did they
Yeah,
biopsy people?
I believe that they're biopsies. So let me double check
That's
quickly.
pretty cool. I mean, there's another thing about choosing papers, like sometimes it's great to choose the papers of like the rare things that people don't often work with, right? So like that's why like at the beginning, like reading all those rhesus macaque papers are really cool because like who works with rhesus macaques, right? Like that's like a rare chance of like finding information. Similarly, like who biopsies lymph nodes in these in patients with COVID-19, right? There's something
Yeah,
nice about that data set.
I mean, yeah, there is like so tissue banks are like built at lots of different hospitals where
Mm.
patients can donate tissues, especially if you're donating your body science or if even something gets taken out for diagnosis, maybe so there are all sorts of ways that they are uh,
Yeah.
involved. They don't. Oh, okay, right. So I mean, I'm concerned about the ethics statement a little bit, but that's because they don't write about written informed consent directly from the patients. So that's a
Mm.
bit of a oh dear. I'm a bit
Always
concerned about
scary,
that.
yeah.
But yeah, and it is mostly very patient focused on looking at. Uh, so so yeah, I think that. I think I would like to talk a little bit about this, but yeah,
I'd be up for that.
yeah, I
So
can.
is that all we have in terms of candidates? I think that is, right? That was
If
everything.
yeah, that's everything. I mean, if you got any other things that you want to bring up from the Zotero, I can see if I can.
No, I mean only that that we should link the feline coronavirus paper. I'll do it later. Link it to the other protease paper that we have in the Zotero.
Yeah, I
It's
put
under.
it in the structure section of the Zotero rather than the treatment ones, but and
Mhm.
left a little note for because I really did feel like I couldn't call this a treatment paper because there wasn't really enough
Sure,
there.
sure.
But it did have some fascinating structural stuff that I thought was quite interesting.
Yep.
And
Yeah, I was
linking that
just
up,
saying that like we have something about protease. So like next time if we choose to go down the protease route, right, we can do that. Um, there wasn't really anything. I mean, I guess we still haven't talked about the whole whole virus vaccine, the CanSino one. They
yeah.
have the mouse model data on that. I remember that. I thought that was an
So
interesting one to talk about.
yeah, I mean now that the Picovac, which has now entered phase two trials, and there's a Sinopharm one, which also is a whole cell vaccine that has also entered phase two trials. So I mean, if we really wanted to bite off more than we can chew, we could do the entire like clinical from bench to bedside of those. If we really wanted
Ah,
to like
right, right.
compare the primate models all the way up to the phase two studies. So I mean, that would be quite a beefy topic to look at.
hmm
So
And that's, yeah, I feel like that's everything that we have. There's some aerosol stuff, meta research.
mm,
Yeah, I guess if we do the conformational, we can also do the spike cryo-EM. That's
yeah,
sort of like the two things that we do together.
yeah,
Uh, the one with the two distinct conformational states and the spike.
yeah. I mean, I feel like because I came in here thinking like maybe something more structural, maybe more cell life cycle like. So
hmm
uh, actually, the structure of the M protease and the feline coronavirus, I would be kind of interested in tackling those.
Okay. Okay, so we have so protease. Um, oh yeah, because that also scratches a bit of the structure, right? Is
Yeah,
that one?
scratch a bit of structure,
Yeah.
and also it's something that people have been talking about in the news. So it means that people
Mm.
are quite interested to hear about how uh, this potential treatment works, and then it's an opportunity to talk about structure, about
Another protein from SARS-CoV-2.
yeah. That's kind of
Mhm.
where I was thinking about going with that. Um, or we,
That's pretty
yeah,
good. I like that. Yeah. Or we go down the immunology route. I mean, immunology is also being so that immunology would be this one or
yeah.
the.
So I guess the BCL5 and the ORF9 ones, they both speak to that idea of well, the immune response is impaired, so that could be
Mhm.
explained, and that I guess ties into reinfections as well. So I guess
Yeah, there's
that
two different ones. We haven't like last week we basically did one that was a bit more on the structure side. Maybe we'll switch. I think we should do an immunology one.
okay, yeah, we'll do that.
Um,
Yeah. Uh,
so should we do BCL, do you want to do follicular or do you want to do the ORF9? What's the older one? Are they
okay,
when are
let's
they
see what the
published?
yeah. Well. check
This
out.
one's October 1st, 2020. That's the future.
Okay,
So
right.
this one's super new, the follicular helper cells.
Yeah, it's available since the 19th of August, so that's been pretty.
The Orphan is just a preprint.
Yeah, the Orphanine is just a preprint, so we will have a chance to talk about that later if necessary. So we can go into
Yeah,
BCL.
and they might clean it up a little bit too at that point.
Yeah, and yeah, I think so. Yeah, I think BCL6. We were won over by the pictures and the whole like germinal
Yeah,
centers.
flipping back to immunology, I think would be nice. I think it's nice to intersperse them. I mean, that was always the thing with pathogenesis, right? Is that
Yeah,
like you kind of have to understand both sides. So
yeah,
in our journey to understand this, we're sort of refreshing ourselves about these topics back and forth.
yeah. No, I think so. We'll definitely pencil that in. It's the paper which we're going to go with in the end is going to be let's see, transition this loss of BCL6 expressing T follicular helper cells and germinal centers in COVID-19. Um, so yeah, that. So if you're watching at home, that's what you need to study up for next week. Tweet us your opinions on it, and we'll talk about it on the show.
Hmm.
Yeah, and this is quite an interesting paper, and I quite there's lots of good things about it. So there's going to be
Yeah,
a lot to talk about.
I'm excited.
Yeah, I'm excited too. So yeah, thank you.
So if anyone has specific questions maybe that they find as they're reading it, then feel free to tweet us with the hashtag or with our
Yep.
our tags below. And if not, we'll see everyone next week.
Yeah, we definitely will. So thank you so much for joining me. It's been great talking to you about microbiology and about the
Mhm.
coronavirus. So hopefully you can join me next week.
Yep.
Bye.